Hey everyone. So today our speaker will be presenting virtually. He will be joining us afterwards for the reception just outside Bayer. So just like always, there will be a reception with food and drinks downstairs outside. So if you want to join us, please do, whether you're watching this in person here or on the YouTube live stream. So our speaker today is Paul Hime. He is an instructor in the Department of Genetics and the McDonald Genome Institute at Washington University School of Medicine in St. Louis. Prior to joining the School of Medicine, Paul earned a BA in biology from Washington University in St. Louis, a PhD in evolutionary biology from the University of Kentucky, and completed postdoctoral training at the University of Kansas Biodiversity Institute and Natural History Museum. Formerly an amphibian and reptile keeper at the St. Louis Zoo, Paul's research interests span structural biology, phylogenetics, molecular evolution, and biodiversity genomics. His current research focuses on computational approaches to understand the relationships between 3D protein structures and enzymatic functions with applications for precision human medicine. Outside of the lab, Paul and his wife enjoy sharing their love of hiking, rock climbing, gardening, and natural history with their 3 children. So please welcome today's speaker, Paul Hime.
Thank you so much. It's just a delight to be here presenting back at my alma mater here at WashU, and walking the the halls of Rebstock this afternoon has really kind of been evocative for a lot of the really formative experiences that I had here, and it's just marvelous to be back. So thank you to the LEC and the seminar organizers for the kind invitation. And also happy equinox. It's just awesome that you were able to deliver up such wonderful weather for this. So yes, my name is Paul Hime. I'm a new faculty member at the School of Medicine working in the McDonald Genome Institute, and I'm excited to discuss some of my research at the interface of biodiversity science, functional genomics, and precision human medicine. A lot of my research interests have largely been driven by curiosity about biological complexity that I observe in nature. And from the earliest ages, my parents really fostered a deep love and appreciation for the natural world. And for me, that's really been a catalyst driving the curiosity that informs the questions that I ask about the world around us and what we can consider the most complex structure in the solar system, Earth's biodiversity. And I'm going to discuss some of my paths from WashU biology as an undergrad to now my lab's work at the School of Medicine. And I'm going to highlight and come back to this triad, if you will, of kind of a lot of my research questions being informed by and driven by curiosity about complexity that I observe in the natural world, and the inevitable consternation that I seem to find myself in when I realize just how little we actually understand about a lot of these phenomena, and this feeling of, wow, that is such a cool observation, but wait, we really don't understand, fundamental aspects about the complexity underlying, this observation or that phenomenon. And I think importantly, that has really driven me in my scholarship to attempt to, seek out disparate information from other fields and try to integrate that together as I try to tackle questions that are largely driven by my curiosity. And in many cases, much of my research has been formed through these questions, which are largely applied, but I have found myself just being really fascinated and motivated by trying to use that as a springboard for probing more general phenomena in biology. And I'll discuss some ways today in which I view the study of Earth's biodiversity as fitting hand in hand with some of the emerging methods that are being developed not only for genome sequencing and model and non-model organisms, but also for computational approaches to understand a lot of this biological complexity. So my research was really formative here as an undergrad at WashU. And I remember early on in my freshman year, walking into Rebstock Hall and seeing this amazing display of vertebrate osteology, which I'm so pleased to see is still kind of gracing that entrance hallway. And as someone with a lifelong interest in herpetology, the study of reptiles and amphibians, that particular specimen just really hooked me early on from coming here. And, really motivated me to say quite bluntly, I want to study that, right? That giant salamander, that is just incredible, right? And throughout my undergrad, I was really fortunate to have the privilege to work with just a huge number of really dedicated and inspirational researchers here. I was advised by Dr. Jonathan Lossis and was fortunate enough to conduct 2 summers of fieldwork in the Greater Antillean Islands on Jamaica and Hispaniola. Working with his grad students there, I was actually on an expedition to the Dominican Republic that successfully sampled all of the several dozen Anolis species there, and just really had my eyes opened to the layers of kind of ecological complexity, and was really curious about all of the phylogenetic relationships underlying a lot of that complexity. I was also in one of the early cohorts of the Tyson Research Center Summer Scholars and was able to conduct projects on kind of the spatial ecology of fence lizards, working with John Parks. And then in my senior year, had an extremely formative research for credit experience at the St. Louis Zoo, where I worked as an intern right when they were beginning a lot of their really impressive work on hellbender salamander conservation. And throughout my undergrad, I was exposed to kind of the full gamut of biological organization, but it was really at the more macro scales, the evolutionary questions, the ecological questions that drove me. And I think as I reflect back on that time, I find myself saying, wow, so many times over, I just didn't get it, right? My cell bio classes, a lot of my, biochemistry course preparatory work just really, I think, took an appreciation of the fractal nature of complexity in nature to realize the interconnectedness across lots of these levels. And today I work at very micro scales, protein evolution, a lot of cellular phenotyping that I'll discuss later. But I think a big foundation of this was just kind of the holistic introduction to biology that I received here at WashU. So as I mentioned, I did WashU undergrad. I stuck around for 3 years and worked as a technician in a couple different labs, applying my interests in reptile and amphibian husbandry to develop captive breeding protocols for Anolis lizards. I also did about a year of moss transformation, where I kind of cut my teeth on a lot of molecular lab techniques, and then pursued a career in zookeeping. So I finally landed what I thought at the time was my dream job, and in many ways was, as a keeper at the St. Louis Zoo. And there I had the privilege of implementing a big amphibian exhibit. It's called Awesome Amphibians. It had a penetrance of over 2 million visitors throughout its duration. And we showcased a lot of the diversity of extant amphibians and tried to highlight some of the ways in which human activities are leading to imperilment of many of these amphibian species. And a lot of my fascination with amphibians at the zoo led me to try to tackle some applied questions as a keeper. And that research drive led me to realize that doing that sort of research on my weekends and at night after work just was not really a tenable solution. And I was motivated to join the PhD program at University of Kentucky, where I worked with David Weisrock on aspects of salamander and amphibian evolution, and later to pursue postdoctoral training at the University of Kansas, where I worked with Rich Glore and many of the other curators, kind of helping act as a liaison between students and PIs and the genomic data that they wanted to collect for their ongoing projects. And a lot of this led me to realize that these applied questions about species conservation were really also analogous to, at the time, a lot of my interests in human health and precision medicine, due in no small part to the SARS-CoV-2 pandemic, which we've all experienced. And I was fortunate enough to join WashU School of Medicine, where I am now getting to work with just a really awesome set of researchers tackling questions at the interface of biodiversity genomics, computational biology, structural biology, and precision medicine. Now, I should acknowledge this amazing creature here, the hellbender salamander, for me was really my gateway into genomics. And as a reptile and amphibian keeper at the St. Louis Zoo, fundamentally, we wanted to breed lots of these salamanders so that we could release them back into the wild. So they're rapidly becoming extirpated across Eastern North America, where they historically lived. And again, the consternation part of kind of my approach to doing science was it's really hard to tell males from females. How are you going to successfully captive breed a species for which you may have to wait 8 years for them to become sexually mature, at which point you can then make a pretty bad guess based on external morphology. Also, we observed that hellbender salamanders from different river systems outwardly looked different. To what extent did those phenotypic differences among populations reflect underlying genetic variation across the range, and led me to ask questions about how many species of hellbenders are there. And again, these were kind of what I thought were fundamental questions. And I was like, surely we know this, right? And, the realization that there were still kind of big knowledge gaps in the way of applied species conservation in this case led me to dig into the literature and attempt to approach this in a scholarly manner. So one of the first research questions that I tried to tackle in hellbenders, and actually began a lot of this research in Alan Larson's lab here at WashU while I was still a zookeeper, involved trying to find a genetic sex determination locus for hellbenders that we could use as a diagnostic to identify males and females in the zoo's captive breeding population. And unbeknownst to me, this was a profoundly difficult question at the time. Non-model organism genome sequencing was just at its advent. And additionally, hellbenders have a genome that's about 18 times larger than yours or mine, at about 56 gigabases. And throughout my PhD, I worked to sample known males and females, to develop sets of anonymous genome-wide markers, and then to computationally compare sets of markers from pools of males and pools of females. And I was able to identify several sex-linked markers that were diagnostic about sex in this group. And interestingly, if you reflect back to that initial giant salamander skeleton on display here on the second floor of Rebstock, also realized that these markers were deeply conserved. And so the sister taxon, Andreas, to the hellbender salamanders, which are the Asian giant salamanders, are also a species of conservation concern. And I thought it was really remarkable that we see this really deep conservation of the same sex determination loci in this deeply divergent species. And so this applied question led me to try to ask some more general questions about the evolution of sex determination in salamanders. Their sex determination systems of either male or female heterogamety are much more labile than we think about those traits as being in, say, avian or mammalian systems. But having delved into the world of amphibian genomics, and also largely motivated by childhood spent catching frogs and salamanders in creeks, and my time working as an amphibian keeper and, in a large sense, ambassador at the St. Louis Zoo, another of my projects here wanted to start looking at the species boundaries in hellbenders. And so simultaneously, I worked with a big team of collaborators across many institutions in the US to do range-wide geographic sampling of hellbenders to tackle questions about how many species there are. Hellbenders are aquatic salamanders. They live at the bottom of rivers. The rule of thumb is if the boulder is too big to lift, that is where the hellbenders are. And so I was able to team up with a lot of people doing field work on hellbenders and amass a collection of about 300 tissues from across the range. Now, hellbenders are riverine. They live in these river and creek systems where you might walk 10 miles across a ridge into a different watershed, but the hellbenders on either side of that might be separated by literally hundreds of river miles. And so there's the potential for really deep geographic and phylogenetic divergence arrayed in a really tight portion of the landscape as the crow may fly, and sets the stage for potentially cryptic lineages of hellbenders here. And sure enough, when we applied many of those same genome-wide markers in a population genetic context, we were able to elucidate 4 primary genetic lineages of hellbenders across the range, which we think may correspond to actual species level entities. And we also revealed that there is additional level of substructure within many of these. And a lot of these lineages of hellbenders now are being considered potentially as functional management units, and we're in the process of kind of finalizing a lot of the formal species descriptions for these. So it was very exciting that kind of a lot of that early interest in amphibian evolution led to what I think were some pretty fundamental filling in of knowledge gaps about hellbender salamanders. As I mentioned, I was really interested in extant amphibian diversity, and there are 3 orders of amphibians. One of the least known is the caecilians, these limbless, tailless, largely fossorial amphibians. They are really spectacular and very poorly known. One of my favorites, the salamanders, has a hotspot right here in Eastern North America. And there's, little more than an order of magnitude, greater number of species of salamanders than there are caecilians. And of course, one of the most recognizable, the frogs and toads, of which there are well over 7,000 species described. And these 3 orders of amphibians share an evolutionary history more than 300 million years in the past. Yet today, the interordinal relationships among these groups of amphibians were still pretty murky. So some studies claimed lots of different relationships among these phylogenetically. And so another of the projects that I tackled in my PhD research was trying to amass genome-wide protein coding markers with which we could tackle questions about deep and shallow scale relationships among amphibians. As a very imperiled group, understanding the evolutionary history has big implications for how we attempt to leverage efforts for amphibian conservation. So to get a good amphibian phylogeny, we wanted to have really good sampling of taxa. I worked with a big international team of over 20 collaborators to amass sampling of nearly every family and subfamily level lineage of amphibians, and also worked to computationally design a set of about 350 nuclear exons, which we could capture in parallel out of all of these really diverse and deeply divergent amphibian species. This, again, right, I've got the curiosity, right? What are the relationships among amphibians? The consternation of, oh, wow, like we really don't understand that. And it's going to be extremely difficult to get genetic markers that will accurately reveal this. And then the scholarship of, okay, now I've got to learn computational biology. I remember my PhD advisor saying, hey, man, this thing called your terminal on your computer, and you and your terminal, you're going to be real close real soon. And wow, what an understatement that was. And so also really began a big push into the genomic and transcriptomic world of non-model organisms. And so my collaborators and I sampled genomic resources from a variety of amphibians, did comparative genomics to identify conserved markers, which might be informative at the deep and shallow scales of divergence here, and then designed a high throughput sequencing system on, at the time, some of the kind of state-of-the-art sequencing platforms to really readily sequence hundreds of protein coding genes for hundreds of amphibian species. And with this, we were able to really, I think, persuasively demonstrate that frogs and salamanders are sister taxa to the exclusion of caecilians. And this is in spite of lots of individual genes kind of supporting all 3 of these topologies. Additionally, I think we were able to bring some clarity to amphibian relationships in that we showed that many of the previous studies of amphibian phylogeny, which had seemingly generated conflicting results, maybe came to those conclusions because of relatively sparse gene or taxon sampling. So we were able to reconstruct what at the time was one of the first really well-resolved backbone phylogenies that included all the major amphibian lineages. We performed some coalescent-based species tree analyses to generate this phylogeny in spite of a lot of noisy signal across any of these individual gene trees, which we examined. And using fossil calibrations from the literature, we were also able to add on a temporal layer to our phylogeny and uncover a big burst of frog diversification right at the end of the Cretaceous mass extinction event. And so from kind of my early interests catching frogs and salamanders in creeks to later working as a zookeeper, trying to maintain and educate the public about these amphibians. Just thought it was so amazing to be able to then like contribute my small little contribution to our greater understanding of amphibian evolution. So, I've been largely driven on the surface by applied questions and then found myself really driven to try to derive more general insights out of those. And so right now we can genetically diagnose sex in cryptobranchid salamanders. We've revealed that hellbender salamanders may actually represent multiple cryptic and deeply divergent species, and hopefully shined a little bit of light on kind of the roots of the amphibian phylogeny. And in these cases, I think applied species conservation questions were always a catalyst that kind of drove me to continue to push on a lot of these thorny questions. But of late, I would say that an additional catalyst here has been an evolving and deepening desire to understand human disease and the extent to which we can leverage genomic and computational approaches to try to tackle really pressing questions in precision human medicine. And I'll switch gears a little bit here and start to talk about some of the collaborative research that my lab and other labs at the McDonald Genome Institute are leading, kind of at the interface of functional genomics and precision human medicine. And so for a long time, and I remember when I applied to WashU, right? WashU was one of just a very small number of schools that was involved with the initial efforts to sequence the human genome. And at the time, I'll admit, a lot of what I just said was totally lost on me, right? I had this notion that humans have DNA, all animals have DNA. It codes for RNA, which encodes proteins, and proteins make up our body. But I didn't really get, and I think I'm still learning about the ways in which our genomic information is really relevant to all aspects of biology and precision medicine in particular. And here by precision medicine, I mean kind of what for a long time was the pipe dream, right? Of if we can truly interrogate the genome of an individual and be able to make predictive statements about either their proclivity for different genetic disorders, or in those cases, how we can actually not only treat patients, but tailor our therapeutics and treatments to that individual's unique genomic sequence. I think it's a just tremendously exciting time in genomics and precision medicine that we truly are on the cusp of a lot of these, long-standing aims. And so let's kind of walk through what this might look like, right? And, I want to certainly highlight the kind of integrative role that many of the research centers at McDonald Genome Institute are lending towards this big initiative, right? And so we've got a patient, and it's really well known, that almost trivial nowadays to generate a genome sequence for individuals, right? I mean, we do this dozens or hundreds of patient samples a day. And from this, we can generate a list of variants, right, that differ from this patient and our set of reference genomes. Cool. Well, what does this actually mean, right? You're not going to hand this variant list to the patient and say, okay, now figure this out, right? So that's what we're trying to do. And it's certainly a far cry from having the genome sequence and a list of variants to being able to say, hey, here's the functional impact or lack thereof of these variants, right? And so one of the labs and lab groups with which I collaborate most closely at the MGI, the McDonald Genome Institute, is denoted as 5, right? So functional imaging for variant elucidation. And here, this lab group is asking questions about which genetic variants in our genomes actually affect cellular phenotypes, right? So we can imagine that if we know of variants that affect cellular phenotypes, it's maybe not too much of a stretch to then make predictions about how these may impact organismal phenotypes and thus different disease phenotypes, right? And so we want to also understand the complexity underlying this, right? So which variants may interact and under which environmental contexts to produce disease states, right? So we've got kind of the functional portion of this equation. We've also just opened a brand new mass spec core there, so MTAC, And here we have ready access to lots of just amazing and cutting edge approaches for structural elucidation of proteins and different metabolites, asking questions about which portions of proteins might we target for therapeutics, right? So say you've got a variant in a protein sequence that leads it to have a different shape than the kind of wild type or normal protein would. Okay, well, maybe we can identify structural aspects of that we could design either a small molecule to kind of remediate those functional impacts. Maybe we could design a custom protein that's new and doesn't exist in nature that could go in and prop up and kind of correct the shape of that defective protein, right? And then I think another big part of all this is the notion that we can leverage the kind of trillions and trillions of years of cumulative evolution over the last 3.8 billion years of the branching history of life on Earth, where evolution now has for billions of years kind of been experimenting, if you will, with what are combinations of genetic variants that work well together, which ones don't, right? And so we're attempting to ask questions like, well, which variants are existing in the human pangenome? So almost a genomic natural history survey of human genetic diversity. What's the evolutionary history of these variants? And how much co-evolutionary constraint may exist between different proteins that we could leverage to try to target really precise therapeutics and diagnoses for patients? And part of, again, I mentioned kind of this pipe dream that I think might actually finally be within our grasp is now trying to connect patients' causal variants to precision treatments. And so we've all, I think, kind of been aware of or almost indoctrinated in this idea that the central dogma here, right, that DNA often encodes RNA, which encodes proteins, which through interactions with the environment result in phenotypes, right? And we can think about phenotypes all the way down from molecular phenotypes to cellular phenotypes, organismal phenotypes. And we know from the last 20 plus years of genomic research in humans and non-model species that there are millions of genetic variants that exist in populations. And some of these are in protein coding regions and may affect protein structure or function. Others seemingly have very little effect on function. And if we can understand which of these variants might lead to or be implicated in disease phenotypes, then we can start to design improved diagnostics and more precise patient-centric treatments. But fundamental to this effort is our ability to assign function to specific variants, right? And so there's a really, robust literature of how we've gone about this as geneticists and more recently genomicists from a lot of the classical kind of model organism experiments. But I think one of the most exciting of the last, a little less than a decade are experimental approaches, right? Where we can now leverage genome editing technologies, which now are, kind of being revolutionized by advances in CRISPR and a lot of the very targeted and very precise genome editing technologies that are just, really flourishing today. So taking experimental approaches such as deep mutational scanning, where we can ask just, amazing questions like, what happens if we experimentally change each amino acid residue in a protein to each of the other 19 naturally occurring amino acids? All right. Which phenotypes, though, are we going to use as proxies for this? And here I'm depicting a graphic where on the horizontal axis, we've got an imaginary 25 amino acid long protein. And here we're showing the functional impact of substituting each of the other amino acids for kind of the wild type, if you will. But in order to have information and make inferences about function, right? And function is a very kind of broad and messy term. We need to ask, well, function relative to which phenotype? If any of us have been paying attention to biology, we know that interaction is really at the heart of biological complexity, right? And interactions among these different variants are almost certainly important, but it also complicates things, right? And so we get this just combinatorial explosion when we think about even testing all pairwise combinations of potential deep mutational scanning variants. And then we get up to trying to, assay a 500 amino acid residue long protein and looking at even all, 1 through 20-fold combinations there. Okay, well, rapidly, we've exceeded the number of particles in the observable universe, right? And so this is still, I think, one of the big challenges for these sorts of efforts to link genotype to phenotype and link phenotype to disease phenotypes in human medical systems. But a lot of emerging methods do allow us to create many variants in parallel and to use some really slick assay techniques to assay a lot of these phenotypes in parallel as well. All right, and parallelization here is really the key to doing any of this in a high throughput manner. So through the 5 lab at McDonald Genome Institute, they are really kind of pushing the boundaries of deep cellular phenotyping, right? And classically trained as a ecologist and an organismal biologist, I thought it was just remarkable when I started learning about these approaches that, just in the sorts of ways that you can observe a population or individuals from different species and kind of by eye pick up phenotypic differences, and certainly our perception as humans and the way in which we're trained to do biology influence what we think of as a phenotype, right? But we can do this for cells too. It blew my mind when I kind of realized what was possible here. And so here I'm just denoting, 3 different staining techniques for the same set of many, many cells here, right? And so we can differentially stain for either nuclei or certain proteins like tubulin or certain organellar components like the mitochondrion. And of course, we think, falling back on some of my organismal training, yeah, it's painstaking, right? To like be under the microscope with calipers trying to measure, different either morphometric or color phenotypes under a microscope for multicellular organisms. And here we're trying to do it for cells, right? And so automating a lot of this is key. And a lot of the kind of mask tracing approaches are allowing us now as a field to start doing automated phenotyping, right? So we can differentiate cells from each other, and we can actually assay dozens or hundreds of unique cellular phenotypes from a single imaging run. Simultaneously, we can do this across, thousands or even hundreds of thousands or millions of cells and just generate insane amounts of deep cellular phenotypic data for cells, right? And just some examples here of ways in which we can in parallel stain and in other ways interrogate different aspects of the cellular phenotype. And for me, I just thought it was so remarkable that a lot of my kind of organismal biology training to pick out differences among populations or individuals could be applied at this, from my perspective, very, very micro scale. So that kind of covers like the phenotyping, right? But in that precision medicine pipeline that I outlined before, well, that was only one part of the equation here, right? And so we also want to start understanding the effects of variants and kind of substituting in different genetic variants and its effect on cellular phenotypes, right? And I think one of the areas in which the 5 labs at MGI are really kind of pushing the envelope here is in being able to leverage a lot of the parallel genome editing technologies that CRISPR and related breakthroughs have really ushered in, right? And so I just find it amazing that now it's almost routine to be able to generate a mutagenic library, right? Where we have different perturbations to the genome, a perturbogen library, if you will. And then in parallel, we can transfect these variants into living cells. In this case, we kind of have a cartoon depiction of human cells, right? And a priori, we don't necessarily know what the impact of these are going to be, right? So we could do this in a hypothesis-driven manner where we have, predictions of if we put in this amino acid in this one protein, well, we have, reasons to suspect that it would lead to the following phenotypes. But also, we can do this almost agnostically, right? And just let the data kind of tell us which variants are important for which phenotypes. If we can go about this in a clever enough way to extract those features from this really information-rich data. And so here in parallel, we can infect millions of cells with different genetic alterations, and then be able to do the deep cellular phenotyping to try to sort out, quote unquote, normal cells from perturbed cells. And one of the key parts here is then being able to segregate out what are our perturbed cells. We can readily sequence them to now start to understand, okay, if I put in the following amino acid changes here, we get the following phenotypes, right? And so now we're starting to get some legs. We can kind of start to connect particular changes in the genome and the ways that they have functional impacts on phenotypes that might be relevant for human disease. As an evolutionary biologist, I was always just fascinated with the concept of convergent evolution. And you see this, of course, replicated across lots of organismal systems, right? So, of course, one of the classic examples is kind of the repeated evolution of communities of anolis lizards, where they've now partitioned the available niches into species which comprise different ecomorphs, so they kind of independently converge on similar morphologies when they evolve in different ecological conditions. Similarly, I just think it's fascinating that you can take that down to the protein level and have examples of, and here, one kind of classic example is what we call the catalytic triad, right? So many proteins are enzymatic, right? They catalyze a reaction. They change one molecule into another molecule. And to do that, there are particular configurations of amino acid residues in the active sites of these enzymes that are particularly effective at catalyzing these reactions. And time again, over the evolution of life on Earth, different proteins have independently hit on combinations of, in this case, this catalytic triad of an acidic residue, basic residue, and a nucleophilic residue. And they can kind of generate this like charge transfer, which then can lead to bond breaking and bond formation in the substrates on which they catalyze these enzymatic reactions. And this catalytic triad, evolution has tinkered with this sufficiently, and now we can see it borne out across 23 different superfamilies of homologous proteins. And kind of the notion here is that evolution can substitute out the particular amino acids that might comprise the acidic or the basic or the nucleophilic residues here, but we've got this really conserved convergent triad of residues, which in the primary structure of the protein may be very far away from each other, but in its tertiary 3D folded structure, they come together to make this, almost magical, this like really amazing catalytic core in these enzymes, right? And so I was just fascinated when I learned about this, that here is potentially this like predictable feature of protein evolution, which we can use not only for specific applications in say protein design or human medicine, but maybe to start getting at more basic features of protein evolution. And so kind of to conclude here, I'll touch on some of my emerging work that I'm doing collaboratively with researchers at the MGI on attempting to use kind of computational approaches to also inform the ways in which we can leverage information about the 3D structure of proteins to start to get at what are the functional impacts of different variants on organismal disease phenotypes. As a phylogeneticist, I was really well trained in performing multiple sequence alignment, right? And so if you think about taking a particular locus in the genome, right? It could be a homologous protein that's shared across many organisms, right? And painstakingly or not, you can sample them from a variety of members of the tree of life. You can use computational algorithms to essentially stack these up, right? And so each row in this is depicting the protein sequence of a given species. And then the columns are essentially evolutionarily homologous amino acid residues, which are shared among these distantly related organisms. And we can see patterns of sequence conservation here. And if you've got a collection of organisms that are separated by 100 million years of evolution, and every single one of them has a glutamic acid, at site 642, for instance, you got some pretty good clues there that might be especially relevant to the functioning of that protein. And more generally, covariation in the ways in which these residues evolve can hint at functional relationships and functional constraints among different regions of the protein. Which kind of brings us to the notion that we can use information from the tree of life, right? So here we're actually tapping into the biodiversity genomics that we can now scrape together either from existing databases or de novo to start to reveal features of proteins, right? And so here I'm just depicting, along the linear length of this protein, different non-proximate residues are actually proximate in the 3D structure, colored here in blue, green, and red. And many of these different protein sectors are very functionally constrained. They co-evolve, the sites in them co-evolve. And many of these are deep in the core of these proteins. And so kind of in the middle space-filling rendering of this particular protein, we fleshed out all the non-sector residues here. And you can see that these sectors in reality sometimes form connections between the surface of the protein all the way down to that kind of embedded catalytic core, right? And so here we're actually able to leverage biodiversity genomics and the information accumulated throughout millions of years of evolution to start to say something about the functional relationships among sites in a protein. And now, thanks to breakthroughs in kind of artificial intelligence approaches to determining the 3D structure of proteins without crystallography, certainly leveraging a whole wealth of crystallographic information. But if you extend kind of the coevolutionary constraints that we observe in protein sequences, within the last year or two, there have just been revolution after revolution in our ability to finally answer kind of these grand challenges of how do we go from the primary linear structure of amino acids in a protein to being able to actually accurately predict its folded 3D structure, right? And this is just really ushering in whole new areas of structural biology that we can leverage for really, I would posit, like just about any aspect of modern biology, right? And so one specific application of this is many labs at the med school and at MGI are very interested in what we call these tier domain proteins. And these are toll interleukin receptor proteins. They have this conserved tier domain, and these are conserved deeply in the phylogenetic history of organisms on Earth. They're found in animals, plants, bacteria, archaea, even some viruses. And these tier domains mediate cell-cell signaling. They're implicated in a lot of immune responses. And fundamentally, were often, originally thought to be structural domains, but recently have been implicated in NAD hydrolysis. And so there are these NADases that were sort of hanging out cryptically under the radar for a long time. One example of this is SARM1, a nuclearly encoded mitochondrial interacting protein in the human genome that has a tier domain and is implicated in a lot of molecular, or rather disease phenotypes like neuropathy and axon degeneration. And this tier domain in vivo can catalyze reactions on NAD. So NAD is this like really fundamental metabolic small molecule. It is catalyzed in complex 1 of the mitochondrial electron transport chain and is really ubiquitous in its importance across all life. And of course, as I alluded to earlier, like the catalytic reactions enzymatically on really any small molecule in general, are kind of dictated by the ligand's adjacent protein microenvironment. And we've been examining a specific case here with tier domains, and I'm working with a set of 309 tier domain containing proteins that a collaborator, Jimmy Weegley in the med school, has been studying as part of a microbiome project. By the way, a little plug for Jimmy. He's going to be presenting at the MDive STL conference coming up through the LEC soon. So be sure to check out his talk at River Camp in a couple weeks here. But these NADases in the tier domains don't just generate one product. They generate a variety of metabolic products. And we want to start linking kind of the ways in which the protein structures can influence not only are these tier domain proteins catalytic or not, but how can we potentially predict just from the ligand-specific protein microenvironment which of these different catalytic products are going to be generated or under which conditions? Kind of how do we do this, right? So we can, from primary sequence, predict these 3D structures of these proteins. We can also use computational approaches to identify pocket regions that might be catalytic, and we can kind of confirm that by doing computational docking of particular small molecules into these pockets. We can also do structural comparisons here. And so don't be fooled. This is not a phylogenetic tree. This is actually a dendrogram showing hierarchical clustering of our different 309 tier proteins based on their 3D structure, right? And so things that are sequences which are closer together on this dendrogram have more similar structures. And here I've highlighted that a subset of these structures are active, denoted in blue, whereas many are actually inactive, which is, I think, why these tier domain proteins were able to fly under the radar for so long and thought to be purely structural. There's a couple ways that we can extract features from this to start leveraging AI approaches to learn more about the ways in which protein structure is impacting its function. I won't belabor this too much, but essentially we can do geometric sequence alignment, right? So we can start to line up different homologous proteins and see which parts are conserved. We can see how their catalytic residues either are or are not concordant in 3D space and start to populate some of our AI models with these sorts of data. We can also just take the totality of protein families and align them in 3D space. I, again, won't belabor the slide too much, but suffice to say, we can now start to categorize in 3D space which residues are actually proximate to other residues from very disparate proteins, and how can we extract features from this, which we can start to use to model enzymaticity, product diversity, all with an aim towards understanding fundamental aspects of protein structure and function, with an aim then towards improving diagnoses and therapeutics for precision medicine. We're also expanding this out, right? So there is just a wealth of bacterial information from reference genomes and metagenomic sequencing. And so we're now expanding this work in tier domains to a set of about 12,000 tier domain containing proteins, which have been generated. And now with the protein folding revolution kind of unfolding, if you will, in front of us, the community is up to a collection of 200 million protein structures now. And so we're trying to find kind of generalized computational approaches to leverage that huge wealth of structural information and start to extract meaningful information about kind of general relationships between protein structure and function. So, in conclusion, my research experiences as an undergrad here at WashU were just absolutely instrumental to my progression as a scientist. And I think of my path through science and trying to discover, new scientific knowledge as this catalytic triad, if you will, of kind of curiosity about natural phenomena lead to consternation that really, we haven't figured that out yet, which then leads to scholarship and attempting to kind of fill some of those knowledge gaps. I think it's just incredible that nature has spent billions of years exploring solutions to these complex problems. and that we can now leverage biological diversity to tackle not only applied, but basic questions kind of at the interface of, biodiversity science and precision medicine. I want to give a shout out to my first student in undergrad right here at WashU, Alejandro Ramirez, who's been cutting his teeth on a lot of these computational approaches. And I think it's just really bringing like some kind of remarkable perspectives and insights to a lot of this work. Want to give a big shout out to a lot of my collaborators in the 5 labs, the functional imaging for variant elucidation. In particular, Willie Buchser, the PI of that lab, who also has a deep interest in evolutionary biology. And it's just been amazing that we found kind of all this intellectual common ground coming from seemingly very disparate parts of the biological spectrum here. And I really got to give a big shout out to my parents for exposing me to nature so early, and to my children who really inspire me on a daily basis with their curiosity. And I hope, not only to kind of instill a love of biodiversity and the pressing need to conserve our biological systems, but also, they are a lot of my motivation for trying to work towards precision medicine, right? So that they and really all of us can, lead the best and most fulfilling lives that we can. And so with that, I'll thank you all so much for attending my talk. And I think I've got a few minutes left before the reception to try and field any questions. So thank you from the bottom of my heart. I appreciate the opportunity.
Thank you, Paul. That was great. So if anybody has any questions, we'll pass around the microphone, and Paul, just let us know if you can hear this okay.
Sure thing.
I wonder, have you tried to look at environmental interactions at the cellular level? So, for example, one of the oldest structural protein ones is sickle cell anemia, which depends very much on the oxygen tension that the cell has, environment of the cell. So, are you trying any manipulating of the cellular environment to see if there's gene by environment interactions at that level?
Oh, yes, that's such a great question. Yes. And I think it really also comes back to kind of this notion that, it's not just that genotype leads to phenotype, right? It's that genotypes interact with the environment to lead to the emergent property of phenotypes. And yes, we're not actively kind of fanning out in that, but yeah, I think there's a lot of potential there.
Thank you, Paul. Really great talk. We've got a question from Ari Miller here on YouTube. He says, such fascinating work on reconciling gene tree, species tree discordance. Did you opt for a target capture approach over a whole genome sequencing approach because of the general difficulty of amphibian genome assembly?
Thank you. Yeah, that's a great question. And yes, we did targeted sequence capture for a few reasons. And so one is our budget was finite, right? And just thinking about sequencing 300 human genomes for a grad student project is like prohibitive for the vast majority of people. A lot of these amphibians, and I mentioned the 56 gigabase genome size of the Hellbender salamander, they're actually kind of right in the middle, right? So some of the other salamanders are up in the hundreds. And yes, additionally, we would have loved to get full genome sequences for all 300 plus species of amphibians in that study. For the purposes of reconstructing the phylogeny, it would have been a huge amount of effort to really then try to distill down to those particular regions of the genome which are alignable, demonstrably homologous, easy to model in a phylogenetic, molecular evolutionary perspective. And so, yeah, 256 conserved exons is like a tiny sliver of those genomes, but was still, I think, information rich enough to be able to fulfill our purposes of kind of reconstructing the phylogeny and getting a range of evolutionary rates with which we could try to estimate the time scale. But yeah, nowadays, probably someone could do that for orders of magnitude less money with whole genome sequence data.
This is a really nice talk. Thanks. I think it's fitting that you worked on hellbenders and then went the protein folding, because I think hellbenders are the animal that looks the most like a folded protein.
I love it. Yes.
I'm curious on looking back at your hellbender work. Even with the cryptic species, I wonder if you can talk a little bit about how you can have some potentially cryptic species inhabiting a relatively small range in the United States, and then other species inhabiting a relatively wide range.
Sure, sure. And I suppose it's fitting also to refer to hellbenders as one of their favorite colloquial monikers, old lasagna sides, which I think really kind of echoes the yes, that protein folding look that they have going on with the lateral folds. I think a lot of the natural history of hellbenders kind of leads me to not be that surprised that there could be deeply divergent species in a relatively small geographic area. So they exhibit very little movement throughout their life, right? They find a boulder at the bottom of the river and they stay there for their 50-year lifespan. You can bring them up or downstream and they hone right back to those rocks, right? So relatively dispersal limited, even in floods, when they get washed downstream, they come right back up. Additionally, the modern day, so the contemporary kind of hydroscapes that they live in have some big boundaries, right? So one of those lineages is on the Kanawha River, the New River drainage in Virginia and West Virginia. And that is literally separated from the rest of the Ohio River drainage by a 7-meter waterfall, right? And so any that get washed down, well, they're going to be unable to hone back up. I think also a lot of the Pleistocene glaciation cycles, the most recent, and I think even glaciation cycles prior to that, really led to like isolated refugia, especially here in the Ozarks with the Ozark hellbender, currently recognized as a subspecies that branches, earliest in the tree of the extant lineages of hellbender. And, my hunch, and we're working to kind of more rigorously test this, is the notion that like they have just been isolated there for so long, right? And the riverine landscape environments are very different. So you get like very different thermal regimes, very different substrates. And so potentially some differential adaptation there, but honestly, I think a lot of it might just be drift that's at play. And so, of course, one would want to test the actual reproductive isolation of these lineages, but yeah, a more dispersal prone species may be expected to have less cryptic diversity.
Okay, we'll probably just do one more question here.
Thanks for that great talk. Not very familiar with the area, but just curious, when you say trace the phylogeny of the different amphibian species using their genome, do you look at specific areas of the genome, or in the bacteria, say they trace with the ribosomal genes or ribosomal RNAs, and I'm guessing they might agree, but if they don't, which kind of is there a systematic way to do it? Just out of curiosity.
Right. Yeah. Thank you. That's a great question. And certainly the phylogenetic markers which we choose, we're assuming are reflective of the underlying evolutionary history that we're trying to trace with that gene tree. And we expect, and would be surprised if we didn't see topological variation gene tree to gene tree, and can kind of reconcile those together into a species tree. But we selected protein coding exons for a few reasons. And one is they're deeply conserved, right? So we have a chance of capturing them in a high throughput manner from deeply divergent lineages of amphibians. If the sequence divergence was too high, we'd have high rates of target capture failure there. As a protein coding locus, they're relatively easy to model, right? So like we can partition them into codon positions and we can apply, various, I think, like plausible molecular evolutionary models there. But yeah, I think the bird phylogeny work where they're actually leveraging whole genomes and partitioning it into different structural regions, right? So here's our exon tree, here's our intron tree, here's our ultra conserved element tree. I think is a really powerful approach to start to understand topological phylogenetic variation across the genome. We didn't do that in amphibians. I would love for someone to do that. I would love to, have someone prove us wrong that, hey, actually, if you look at this bigger picture than just these few hundred exons, here's this more robust or accurate view of the amphibian tree. But yeah, I think it really just goes to trying to select question-specific markers and try to scrupulously build your phylogeny in that manner.
Thank you, Paul. I think that wraps up for today, and we'll see many of you downstairs at the reception.
Wonderful. Thank you all so much. It's been a real delight, and I look forward to seeing you at the reception.